Good evening, folks. Hope everybody is doing okay right now. This is part 2 in my lesson on animal circulatory systems. If you have not already watched part 1, I recommend that you go and do that so you have the necessary background for what I'm about to talk about. And if you are in my biology class, I recommend you hop on Blackboard and get a copy of the handout going with this section so that you have all the relevant vocabulary right in front of you, as well as a summary of the basic concepts we're going to be talking about. So last time we took a look at some general features of animal circulatory systems, talked a bit about the function of circulatory systems and why they are needed, discussed why certain simple animals like cnidarians and flatworms don't really need a circulatory system, and then took a look at open versus closed circulatory systems and the differences among those, as well as what you get when you have a 2-chambered, 3-chambered, or 4-chambered heart in various types of vertebrates. And that's about where we left off. So we had seen that if you're a fish, 2-chambered hearts, single circulatory loop passing through 2 separate capillary beds along the way. First gill circulation, then systemic circulation, but losing a lot of pressure in the course of so doing. Then we have our amphibians and our reptiles, which have double circulation because they have a 3-chambered hearts. They have one shared ventricle, one shared pumping chamber, but 2 separate atria or receiving chambers for blood. That the blood goes out of the left ventricle towards the animal's systemic circuit, delivers oxygen around to the body, and then returns to the right ventricle, where it enters the pulmonary circuit in a reptile or pulmocutaneous circuit in an amphibian to go to the animal's lungs, or in the case of amphibians, lungs and or skin, in order to pick up oxygen, which is then returned to the left atrium. So 2 separate circulatory loops, but only one shared ventricle pumping blood from both of the atria, which means that although we have a renewal of pressure by coming back to the heart, we also have a mingling of oxygenated and deoxygenated blood, which means that we're going to not be delivering oxygen as efficiently as we might. It is not a coincidence that our endothermic animals, that is, birds and mammals, have a distinct 4-chambered hearts. 2 separate atria, 2 separate ventricles, no mingling of oxygenated and deoxygenated blood at all. That we have the right side of the heart servicing the pulmonary circuit, sending blood to the lungs to pick up oxygen, returning it to the left side of the heart, the systemic circuit, to move it throughout the body and deliver it. In fact, before we do anything else, let's kind of just trace a drop of blood. Let's go through the story of mammalian circulation. Birds, we follow the same pathway. So let's start a given drop of blood. Let's begin in the right atrium. This is a loop, it's a circuit, there's no actual beginning point, but that serves as good a starting point as any, because that is the point at which we have deoxygenated blood. It has already made its way through the body and dropped off its oxygen load. So the blood in the right atrium is low oxygen, high carbon dioxide. So that blood in the right atrium is going to start off there. Right atrium is going to contract, push it through the right atrioventricular valve, ATRIOVENTRICULAR, atrioventricular, or sometimes called the right AV valve for short, cardiac medicine. We go from right atrium through the right AV valve into the right ventricle. Right ventricle then contracts, forces blood out through a large vessel called the pulmonary trunk, TRUNK. The pulmonary trunk is one of the 2 largest arteries in the body. The pulmonary trunk forks into left and right pulmonary arteries, which deliver blood to the left and right lungs, respectively. Then they branch into smaller and smaller and smaller arteries throughout the tissue of the lung, eventually culminating in pulmonary capillary beds. And remember, capillaries are where the action happens. They are the vessels that have very thin walls intended explicitly for gas exchange and nutrient exchange. So in those pulmonary capillary beds, the blood is going to unload carbon dioxide, which is then going to be diffused into the lungs to be released from the body, and load up on oxygen, oxygenating that blood for the use of the body. Once that has happened, the blood will be collected in pulmonary veins, which will then all ultimately converge on a right and left pulmonary vein and return to the left atrium of the heart. That's the pulmonary circuits. That's the right side pulmonary pathway to get blood oxygenated in the double circulation of a mammal or a bird. From there, we move into the systemic circuit. Systemic circuit could be said to begin at the left atrium. Blood passes through the left AV valve, the left atrioventricular valve. By the way, in humans, the left atrioventricular valve has a particular name. Well, it's called by two other names. It's called the bicuspid valve. You might have heard of that. B-I-C-U-S-P-I-D, bicuspid, meaning two cusps. C-U-S-P is cusp. The cusps are little leaflets that cover up, they make up the actual tissue of the valve that prevents the backflow of blood. Most of your heart valves are tricuspid, T-R-I, meaning that they have three cusps that make them up. The left AV valve is the only bicuspid valve in the heart. And in human cardiology and anatomy, we often call it the mitral valve, M-I-T-R-A-L. That's an old name given to it back in the Middle Ages, whenever anatomists decided that it resembled the shape of, the big, tall, pointy hat that bishops wear. That's called a mitre. So the term mitral valve stuck. That is incidentally the valve that sometimes in some individuals will prolapse a little bit, P-R-O-L-A-P-S-E. Prolapse means to fold backward or fold in the wrong direction. If you have mitral valve prolapse, or MVP, you will have a little gurgling sound that accompanies each heartbeat. We call that a heart murmur, M-U-R-M-U-R. Heart murmurs are especially common in younger people, particularly for reasons we're not clear on. Young girls. We're also not clear on why, but they're often self-correcting by the time you get to adulthood. If you do have one into adulthood, you'll have to keep an eye on it. It could be a precursor for a little bit of, heart trouble later in life, but it can be surgically corrected if necessary. But for the most part, you can live with an MVP. You might get fatigued a little bit more easily because of the backflow of blood. But, otherwise it's a problem that can be dealt with as long as it's not too severe. Anyhow, blood goes from left atrium through the left AV valve, or bicuspid or mitral valve, into the left ventricle. Left ventricle, by the way, is the most muscular chamber of the heart. It has the thickest walls because it is the one that has to push blood through the entire body. So, out of the left ventricle, blood passes through the left semilunar valve. I don't think I said that before on the other side, but there's a second valve at the exit of the heart. So there are the so-called semilunar valves, S-E-M-I-L-U-N-A-R, which means half moon. They kind of have half moon shape to them. The one at the pulmonary circuit, the one that goes from right atrium to pulmonary trunk, is the right semilunar valve, also called the pulmonary valve for obvious reasons. The one exiting the left side of the heart is the left semilunar valve, also called the aortic valve, A-O-R-T-I-C, given that name because it leads immediately into the aorta, A-O-R-T-A. The aorta is the largest artery in the body. Aorta curves away from the heart. So there's a big bend in it called the aortic arch, A-R-C-H. And then we start distributing the blood to smaller and smaller and smaller arteries. We spread the blood out throughout the body. It eventually goes into systemic capillary beds. There it unloads oxygen and loads up on carbon dioxide. That is eventually collected by systemic veins. Those systemic veins then eventually converge on larger and larger and larger veins, as veins are wont to do, ultimately all emptying into two large veins, the biggest veins in the body, the so-called great veins, known as the vena cavae, V-E-N-A, vena, cavae, C-A-V-A-E. If you're talking about just one of them, it is a vena cava, C-A-V-A. You have two of them, a superior one that comes from above the heart, inferior one that comes up from below the heart. They both converge on the right atrium, and that completes our systemic circuit. So just follow the bouncing ball here, and we'll just go through the path real quick. Right atrium to right AV valve to right ventricle, out through the right semilunar valve, also called the pulmonary valve, into the pulmonary trunk. From there, distributed to various and sundry left and right pulmonary arteries, branching into smaller and smaller arteries till we get into pulmonary capillary beds, where gas exchange happens, take in oxygen, release CO2. go into pulmonary veins, which converge on larger and larger and larger veins until left and right pulmonary veins enter the left ventricle, ending the pulmonary circuit. Now we begin the systemic circuit, in which blood that is now oxygenated goes from the left ventricle through the left AV valve or mitral valve or bicuspid valve into the left ventricle. Did I say left ventricle twice? Left atrium through the valve into left ventricle. Then we pass out through the left ventricle by way of the left semilunar valve, also called the aorta. Aortic valve. Then we go into the aorta. From there, we split off into smaller and smaller and smaller systemic arteries, which eventually empty into systemic capillary beds where we engage in gas exchange, unload oxygen, load up carbon dioxide. From those, we drain into larger and larger and larger veins, eventually converging on the superior above the heart and inferior below the heart vena cavae, and the vena cavae both empty into the right atrium, ending the systemic circuit. That is the pathway of blood for all mammals and all birds. It's a pretty effective way of getting a steady supply of nutrients and oxygen around the body. It is necessary to have that complete separation of left and right circuits in order to sustain an endothermic metabolism. All right, so gotten as far as mammals here. Let's talk a little bit about ourselves, just because I want to tell you a little bit more about how the parts of the cardiovascular system work, and the human heart is the most familiar example, so let's talk about our own. Your heart, fist-sized organ, approximately located right about here in your chest. It's located in that cavity I mentioned last time, the mediastinum, which is between your lungs, immediately behind your sternum, sternum, or breastbone. The heart is tilted slightly to the left. It's not perfectly symmetrical down the middle. About 2/3 of its mass is to the left side, which is why the left lung is a little bit smaller than the right lung in all mammals. When you're looking at mammalian hearts, you will find the atria have relatively thin walls, not as much muscle, because they all need to get blood from atrium to ventricle, which is right there. The ventricles, on the other hand, have thicker walls because they have to force blood further out into the body, and the left ventricle has a thicker wall than the right ventricle because it has to power the entire system, as opposed to right ventricle, which only has to get blood to the lungs, which are right nearby. Now, you know that your heart has a rhythmic cycle. You know that your heart maintains a steady rhythm. This is a really cool thing about the vertebrate heart. Our hearts are autorhythmic, 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 meaning they are self-setting of their rhythm. They set their own rhythm. Now, if you were a squid or another invertebrate, if you're a squid, if you're an earthworm, if you're an invertebrate with hearts, you have what we call a neurogenic pulse. Neurogenic. Neurogenic, meaning that the pulse comes from the nervous system, that the hearts are stimulated constantly by the nervous system to generate the animal's pulse. If those nerve fibers aren't working, the heart doesn't beat. Not so with you. As a mammal, you have, actually as a vertebrate, let's say, you have a myogenic pulse, myogenic. Prefix myo means having to do with muscle. Ergo, your pulse is generated in the muscle itself. Or to put it another way, your heart makes its own pulse. There's a little patch of tissue inside the right atrium of the mammalian hearts called the SA node, the sinoatrial node, sinoatrial. Sometimes doctors just call it the pacemaker. For sure, you've all heard that term. You all heard Grandpa got a pacemaker. Grandpa already had a pacemaker. His pacemaker wasn't working. You gave Grandpa an artificial pacemaker because his heart wasn't setting its own rhythm properly. But if everything's going well, your pacemaker is a little patch of muscle tissue that continuously generates waves of electrical stimulus, causing the hearts to contract and beat. Pacemaker sets its own rhythm completely independent of the brain. The brain can step in. The cardiac nerve can send signals to the pacemaker to stimulate it and ramp it up and make the heart beat faster whenever you're exercising or otherwise need more oxygen flow faster. Likewise, the brain can send signals along a nerve called the vagus nerve, VAGUS. It's one of those cranial nerves I mentioned a chapter or so ago. And the vagus nerve slows the heart rate down. Actually, cool thing to know right now while you're sitting in your seat, your vagus nerve is almost certainly slowing your heartbeat down a little bit, because left to its own devices with no other outside control, the SA node would set a baseline rhythm for the heart of about 100 beats per minute. We don't need that much. So the vagus nerve sends a constant signal to dampen down the heartbeat whenever it is not needing to be that fast. But yeah, SA node generates its own rhythm. If I lay you out on the table and cut you open like an Aztec sacrifice, we'll do that for lab next time, then I can pick your heart up and lift it up to the air, and it would keep on beating in my hands for a minute because it's generating its own rhythm. Now, it wouldn't last long without an oxygen supply, but, you know. All right, so I just told you 100 beats per minute is too fast. What's the right heartbeat? It varies. Actually, there's no one set value for exactly what the textbook healthy human heart rate would be. Most textbooks will tell you that the average healthy adult human heart rate at rest, that is, you're not exercising, you're not excited, you're not doing anything to get your pulse going, and you're in good health, is about 75 beats per minute, BPM, standard measurements. That number will vary a lot depending on what kind of mammal you're dealing with. If you are a mouse, your heart is probably going like 300 beats per minute normally. Tinier animals, remember, lose heat faster. Tinier animals have higher metabolisms. Their hearts have to keep up accordingly. Larger animals tend to have much slower heart rates. We've recently learned that part of the reason why whales can stay underwater so very, very long when they dive is because when a whale dives deep into the water, its pulse falls to as little as like 4 beats a minute, and therefore that animal's not moving blood very, very quickly. Now, terms of the heartbeats, the part of the heart where it is beating, the rhythm where it contracts, is called systole. S-Y-S-T-O-L-E. You pronounce every part of the word. Systole. That is the contraction of the heart. Relaxation of the heart is diastole. D-I-A-S-T-O-L-E. Diastole. So systole, diastole. Those are the two moments of contraction and relaxation. The whole heart does not beat at once. The atria of the heart contract before the ventricles. Why? Because the ventricles need time to fill with blood. It's not going to do them any good if they contract the same time the atria are and they haven't received the blood yet. So the atria contract first, and then the ventricles contract a moment later. There's actually another little part of the heart called the AV node, the atrioventricular node. It's a secondary node located between the atria and the ventricles, and its whole job is to delay the signal given off by the SA node slightly so that the ventricles have time to fill before they beat. The heart has a complete rhythm of contraction and relaxation called the cardiac cycle. When you hear about the cardiac cycle, that's one complete sequence of filling up the atria, contracting them, filling up the ventricles, contracting them. All of that, that's the one pass through the cardiac cycle. That is one heartbeat. Your pulse, of course, is the diagnostic tool we use for measuring this. Your pulse is generated by two things. The sound of arteries stretching as blood is forced into them, but more than that, the sound of the valves opening and closing. You know, your heart rate, when you hear a heartbeat, you expect right? You know that kind of rhythm? Lub-dub, lub-dub, lub-dub. That's actually something that you'll see in cardiac textbooks. Lub, L-U-B-B, dup, D-U-P-P, lub-dub, the rhythm of the heart. The first part, the lub, is a longer, slower stroke. The dup, the second part, is the shorter, faster, sharper stroke. When you're taking a pulse, first part is generated by the sound of blood whacking against the AV valves as they slam shut. And then second is generated by the sound of the semilunar valves closing as the blood exits the hearts. All right. Now, before we get any further into this, actually, I think that's pretty much what you need to know about hearts, right? You feel like you got a handle on hearts? I hope you do, because you can't hear me asking that question. Anyway, you can hear me, but I can't hear you. So, all right. So we get out of the heart. Other part of the cardiovascular system, of course, is the blood vessels, of which you have three distinct kinds. First off, you have the arteries. Arteries are the afferent vessels. They're sometimes called the resistance vessels because they have thick muscular walls. They have to resist surges in blood pressure coming out from the heart. An artery, actually, because of the muscle in its walls, will hold its shape whenever it is empty. It won't collapse whenever it's empty. It'll stay rounded. Arteries are a bit elastic. They have to be a little bit elastic, at least, to accommodate the surges of blood pressure popping through. If they cannot do that, if the artery is rigid, if it cannot tolerate the surges in pressure, that blood is forced faster downstream, which puts more strain on smaller arteries further from the heart, and that can cause them to develop aneurysms, A-N-E-U-R-Y-S-M. No, it's Y-S-M, not I-S-M. An aneurysm, by the way, is not an event. If you say, okay, Bob had an aneurysm, Bob had a ruptured aneurysm is what you're saying. An aneurysm is a structure. It's a little bulge that forms in the wall of the blood vessel whenever the pressure surges into it because there's a weak spot in the wall, causes the aneurysm to pulse in tune with the beating of the heart, and a sudden surge of pressure could cause that aneurysm to pop, which could lead to internal hemorrhaging, and depending on how bad the aneurysm is and where it is, could kill you. So, non-trivial problem. What would make the arteries less flexible? Hardening of the arteries, right? Atherosclerosis, A-T-H-E-R-O-S-C-L-E-R-O-S-I-S, atherosclerosis, hardening of the arteries, as we call it, buildup of fatty plaques of cholesterol on the inside of the arteries, makes their passageway narrower and makes them more rigid, so they're not as easily able to accommodate surges in pressure. Now, I should also point out the arteries are providing the resistance against the flow of blood, and that resistance is how we measure blood pressure. Blood pressure, or BP, is measured by the surge of blood coming into the arteries. When you take your blood pressure, it's two numbers, one above the other one. Top number, you might already know this, is the systolic pressure, S-Y-S-T-O-L-I-C, like systole. The systolic pressure is the highest the pressure rises while the heart is in systole. That is the highest pressure we achieve while the heart is contracting. It's the greatest amount of pressure measured in the artery. Typically, we use the brachial artery in the upper arm in humans to measure that. It's a convenient spot. The lower number is the diastolic pressure. Diastolic pressure is how we usually pronounce it, from diastole, D-I-A-S-T-O-L-I-C. Diastolic pressure is the lowest that the pressure falls during diastole. In other words, the lowest the pressure gets to when the heart is relaxing. So our blood pressure is highest it goes when the heart contracts, over lowest it goes when the heart relaxes. You probably heard your whole life, textbook perfect human blood pressure is 120/80. That is measured in millimeters of mercury, mm capital H lowercase g. This lowercase mm. Millimeters of mercury, named for the device, with the tube filled with mercury that goes up and down in the pressure reader. We don't use a mercury-filled tube anymore these days, but, that's how it used to be done, and that's the standard unit we still measure by. So you've all heard your whole life, 120/80 is the standard textbook perfect human blood pressure. Recent cardiological studies have suggested that's actually a little bit off, that probably textbook perfect human blood pressure is a little closer to 120/75. What affects this? A couple of things. The amount of resistance from the arteries. The more the arteries push, and the more rigid the arteries are, the more pressure the blood will be under. How forcefully the heart is beating, the force of the pulse pressure, as we call it, the distance between systolic and diastolic. The higher that is, the more forcefully blood ejects from the heart. And how fast the heart is beating is obviously a factor as well. Another important factor is the diameter of the blood vessel. This is a thing your body can actually change, because your arteries have muscular walls which allow them to widen and narrow as needed. We call that vasomotion, V-A-S-O-M-O-T-I-O-N. It's controlled by the medulla oblongata of the mammalian brain. Vasoconstriction, V-A-S-O-C-O-N-S-T-R-I-C-T T-I-O-N. Vasoconstriction is the narrowing of blood vessels. Vasodilation, V-A-S-O-D-I-L-A-T-I-O-N, is the widening of blood vessels. Our bodies will constantly adjust the diameters of blood vessels to route blood where it is most needed. Here's the fun fact for you. 75% of your capillaries are empty at any given moment because there's not enough blood in your body to fill every capillary at once. And you know what? We don't need them to. We need them to go where it's most required. If you've just had a big meal, then what you're going to have is more blood flow going towards your stomach, towards your digestive organs, to be there to stimulate digestion and to absorb the nutrients being picked up. You know how you feel all sluggish and heavy-limbed after a big old meal? Yeah, that's because the blood is being routed away from your muscles towards your stomach. That's why they tell you not to exercise after a heavy meal. Now, suppose you are exercising, then we're going to route blood away from the digestive tract towards the muscles. That's going to involve constricting the blood vessels in the digestive tract, dilating the ones in the muscles to open up and let more blood in. So our bodies use vasoconstriction to limit blood flow to places where it's not as much needed, vasodilation to move it to where it is needed. And that, of course, causes concomitant changes in pressure. Widen the vessel, pressure is going to drop within it. Narrow the vessel, pressure is going to increase. And we make constant little blood pressure adjustments that way all the time. It occurs to me, I never answered the question of what's normal human pulse, did I? I should back up on that. I said 75 beats per minute was textbook. Odds are pretty good. Yours is not exactly 75 beats per minute. The normative range of human pulse, which things are usually working okay if you're somewhere in that range, is between about 60 beats per minute and about 100 beats per minute. If you're lower than that, we call that bradycardia, B-R-A-D-Y-C-A-R-D-I-A. Bradycardia is abnormally slow pulse. Sometimes that can happen because something is wrong with you. We all go into bradycardia naturally when we are sleeping. It can indicate damage to the heart. It can also be a sign of solid athletic training. Your well-conditioned endurance athletes, your swimmers, your bike riders, your marathon runners, people like that, will have a lower resting pulse rate because their hearts are stronger and they're getting more squeeze out of each beat. The contraction lasts longer, so they don't need to beat as often. Lance Armstrong, at the peak of his biking career, had a resting pulse of about 32 beats per minute, which for an ordinary person not conditioned like him would probably kill you. Now, other end of the spectrum, higher than 100 beats per minute, you all heard this one, tachycardia, T-A-C-H-Y-C-A-R-D-I-A. That is abnormally elevated pulse. We all get moments of tachycardia when we are exercising, when we are stressed, when we are upset, when we are frightened, when we're sexually aroused. All those things stimulates increased blood flow and stimulate higher heart rate. Persistent tachycardia can be indicative of any number of heart pathologies and can be a significant contributor to high blood pressure, which is why I bring this all up now. You will find in general, male animals typically have lower resting pulses than female animals. Your average adult male animal's resting pulse rate is going to be, okay, I can't give an exact percentage, but let me show you an example in humans. Average healthy adult male resting pulse in humans tends to be between about like 62 and about 76 beats per minute, whereas average healthy resting adult pulse in human females tends to be about 72 to 80 beats per minute. You know, that's the normative range in which we expect things are okay, but your doctor is probably not going to sweat it too much if you're around 60 or if you're just shy of 100, unless it becomes a persistent issue. All right. So, move out of arteries into capillaries. Capillaries are our next type of blood vessel. They are the smallest blood vessels, microscopic, weren't even discovered for a very long time until Antony van Leeuwenhoek, the father of microbiology, you might have heard of him in a previous class, first observed them under the microscope in the 1600s. Capillaries, as I've indicated, have extremely thin walls, only a single layer of simple squamous epithelium thick. That allows us easy diffusion. Allows us? Did I just say allows? Really? My golem? Allows us, precious. Going crazy here in isolation. All right, so, the thin capillary wall allows nutrients to diffuse easily across. When blood flows into the capillaries, we're going to have so-called fluid exchange with the tissue. That fluids are going to exit the capillary, diffusing through, carrying with them oxygen, nutrients, sometimes small soluble molecules in the bloodstream. Fluids will diffuse back out into the capillary, taking away carbon dioxide, metabolic wastes, and anything that needs to exit the tissue. Like, for example, if that tissue is a gland secreting a hormone, that's when it will enter the bloodstream. All in all, about 85% of the fluid that diffuses out of a capillary will diffuse back in at the other end. The remaining 15% or so will remain in the tissue as a clear fluid called lymph, L-Y-M-P-H. Your body has an entire system called the lymphatic system, whose purpose is to collect lymph, filter it, and return it to circulation. The body makes about 4 liters of lymph a day, but it gets recycled and restored to circulation eventually. Now, as the lymph is flowing through its own pathways, through lymphatic ducts in the body, the lymphatic ducts are going to pass through lymph nodes, N-O-D-E-S. Lymph nodes filter the lymph. They contain lots of white blood cells for the breakdown of bacteria and cancer cells and other pathogens that might have been circulating around in the bloodstream. So the lymphatic system is not just for collecting lymph and returning it to circulation. It's also a vital part of our immune systems. In fact, what we call the immune system is not an organ system at all. It's actually a set of lymphatic tissues carrying out a particular response. All right, now, thing I wanted to mention here about all this, the blood, wait, no. Okay, I already mentioned it. Never mind, got lost in my own head. So, blood filters through the capillaries. We engage in fluid exchange with the tissue. If we're going in the systemic circuit, we're releasing oxygen and nutrients into the bloodstream, into the tissue. We're collecting metabolites that need to go somewhere else to be removed. If we're in the pulmonary circuit, we are taking in oxygen from the lungs, and we're releasing carbon dioxide to be exhaled. Now, our last kind of blood vessel, after you leave the capillaries, the blood is going to have to be drained out of the tissue via veins. Veins are very different than arteries. They are much more elastic. They're much stretchier. They're sometimes called capacitance vessels. They have to be stretchier because venous flow is slower. Arterial flow is benefiting from the pressure of the hearts, but by the time you get through the capillaries, most of that pressure has been lost. Therefore, venous blood flow is more sluggish. It's more slower. At any given moment, 54% of your blood at rest is in your veins. And that slower return means that the vein has to be stretchier in order to move along. It also means that any vein below your heart is trying to work uphill and force blood uphill at relatively low pressures. How do we do that? Valves. Veins have valves, unlike arteries, which will close up when the blood tries to fall back down and prevent the blood from moving downward, back the way it came from. Now, veins are getting a little pressure from the heart, but they are also assisted in their action by the movements of the body. As your body moves around, we talk about the so-called skeletal muscle pump, which is going to squeeze the veins and force blood along through them. This is why regular exercise is good for your circulation, because it's improving venous return. We also benefit from gravity assisting any vein above the heart. We benefit from a little bit of suction at the heart end, pulling blood in. And of course, a little bit of pressure from the heart remaining. But venous flow is slower. If you hold still for too long, the stretch on the venous valves becomes more difficult to bear. The blood can begin to pool, and you can experience, pooling of blood in lower extremities, which can result in passing out if you hold still long enough, because too much blood drains from above. By the way, if you want to get a really clear picture of how this looks, of what it looks like when you're experiencing venous pooling, let me just do an experiment right here. We can do this right now. You probably have 2 hands, right? Let's assume you do. If you don't have 2 hands, do it sequentially with whichever hand you've got. All right, take your 2 hands, pick your favorite one, put it down below waist level, put your other one up above your head. Now hold them there for a minute. What's happening now is that as we are doing this, the veins in my arm that I'm holding up are getting an assist from gravity. It's making it easier for venous blood to drain out of my arm. On the other hand, the hand that I have down below my waist, the blood is pooling there more. That hand is hanging down. It's having to work harder against the force of gravity. And while it's hanging there, there's a certain amount of venous pooling that is going on. Still have your hands in position? I hope you do, because in a moment we're going to change them, and I'm going to let you know what to do, and you're going to see a very dramatic example of what I'm talking about here. So on the count of 3, what I want you to do is to lower your hand that's above, raise your hand that is below, and put them together where everybody can see the color of them, or where you can see the color of them. All right, ready? 1, 2, and 3. Okay, I guess in my camera it's not as obvious, but hopefully you saw in yourself that the hand you had below your waist should have been darker in color than the one you had above your head, because the venous pooling was going on there. I'm still looking at them. Yeah, my left hand is still darker than my right. I don't think you can see it here. Left hand, right hand. Can you tell? Yeah, actually you kind of can tell there, huh? All right. So that's the parts of the mammalian cardiovascular system. To finish this out, let's talk about what it's moving around. Let's talk about blood. Gotta say it like that. Blood. Okay, blood. So blood is the mechanical fluid that is circulating around in cardiovascular systems. Remember that blood is a thing you have if you have a cardiovascular system. It is not a thing that you have if you have an open circulatory system, because in that case you have hemolymph instead. Now, in mammals and in most vertebrates, the blood has an iron base, which means that it's got iron circulating in it, and as part of its oxygen collection process. You probably know that oxidized iron is a red color. It comes from the bright red of our blood. You'll find that some other organisms have a greenish blood because their blood contains more copper than it does iron, for example. Spiders, for example, have green blood. All right. And my favorite one, actually, is horseshoe crabs. Horseshoe crabs have blue blood. It's a nice, pretty sky blue color. And incidentally, horseshoe crab blood is a vital medical tool. It is used for bacteriological testing kits. And it's one of the most valuable biological substances in the world, gram for gram. All right, then. So, blood itself, the mechanical fluid that moves through your body. It's got several different functions. Most obvious one, blood is involved in the transport of gases. Collects oxygen from the lungs and brings them to the tissues. Collects carbon dioxide from the tissues, brings it to the lungs for removal. Secondly, circulating nutrients. The blood picks up all of the digested materials from your food, the vitamins, the minerals, the amino acids, carbs, and lipids, circulates them around in the body to the tissues that require them. Thirdly, blood acts to move around hormones and other signaling molecules through our bodies. Fourthly, blood acts to circulate certain vital proteins, including immunological proteins like our globulins that help us to fight infection and other pathogens. Blood also serves to regulate electrolyte and pH levels throughout the body. It acts as a buffer. It acts as a reservoir for certain electrolytes, most notably sodium and potassium. The blood also acts as a fluid reservoir for the body, shifting water where it is and is not needed, accepting water from tissues or diffusing it into tissues. And we don't think about it much, but blood also serves a heat distribution function. Any tissue that's getting more blood flow, gets warmer. And so our bodies adjust the blood flow to accommodate temperature stuff. You want a great example of this? When animals are out in the cold, what happens? They get paler, right? You get paler because your blood is being withdrawn away from the surface of your skin. That you're going to have vasoconstriction in surface blood vessels because that pulls blood away from the skin so you don't lose as much heat to diffusion in the environment. You come back in out of the cold, what happens? You get all red in the face after you've been outside in the cold for a while. That's because your body is opening up those vessels again, vasodilating them, to restore blood flow to tissues that have had less of it for a while. Now, if I'm looking at mammalian blood, and humans are not terribly unusual in this regard, your blood has a couple different components. Mammalian blood is about somewhere around 55% plasma. 54-55% plasma. Plasma is a clear yellowish fluid. Plasma is where vitamins and minerals circulate. Dissolved nitrogen can be found in plasma. Bubbling of dissolved nitrogen in plasma causes the bends if you are a diver and you've been diving for too long. Or not diving too long, rising too fast after diving. You also are going to find in the plasma a number of different clotting factors, proteins that circulate in inactive form to help the blood clot quickly whenever there's a break in a blood vessel. You're going to find a lot of so-called plasma proteins, such as albumin, the same protein found in egg whites, and your various globulins, Globulin. Globulins are, for the most part, immune proteins. They're playing antibody type roles, like gamma globulins are basically antibodies. So, yeah, about 55% of your blood is plasma, that clear yellowish fluid. Now, the rest of your blood, the other 45%, is what we call the formed elements. The formed elements are cells or cell fragments. For the average healthy adult human, about 45% of the volume of blood is going to be a formed element known as erythrocytes, E-R-Y-T-H-R-O-C-Y-T-E. Prefix eryth means red. Those are your red blood cells, or RBCs for short. Red blood cells are the oxygen-carrying cells. They're really nifty. They have sort of a donut-like shape, more surface area to absorb oxygen. They are going to have no nuclei. They have no organelles. Basically, they're just full of sacs of hemoglobin, Hemoglobin. Hemoglobin is the gas-absorbing pigment that both absorbs oxygen and carbon dioxide. So hemoglobin is essential, and by taking most everything else out of the red blood cell, it's basically just filled up with as much hemoglobin in its cytoplasm as possible. Red blood cells wear out periodically. They only live about 120 days, and then they get hemolyzed or broken down. That's Hemolyzed, hemolyzed. They get hemolyzed by the spleen and also by the liver, which when it hemolyzes them, collects a fluid from them called bilirubin, B-I-L-I-R-U-B-I-N, which it turns into bile that I mentioned in the digestive chapter. But yeah, so red blood cells, about 45% of the volume of your blood. They're the ones that are doing the actual oxygen carrying job. You will find adult human males and male mammals in general tend to have a higher percentage of red blood cells. This is definitely a thing. And as a result, male mammals in general tend to have a slightly higher oxygen carrying capacity and slightly higher endurance for the same level of exercise. If anybody lives at higher altitude, male or female, the red blood cell count is going to rise. If you spend a lot of time living in a high altitude area where the atmosphere is thinner, your body compensates by producing more red blood cells in order to try to help carry more oxygen effectively, get more bang for your buck out of each breath. And so if you're living up in the mountains, this will be a thing. There's a reason a lot of athletes train in Denver in the off season. It's because that higher altitude forces their bodies to build up a higher reservoir of red blood cells. And when they come back down to sea level, they still have that heightened red blood cell count for a couple of months. Ergo, they will have heightened endurance for a little while. This is why also, I'm here in Pittsburgh, the Steelers go play in Denver. They go out a week ahead of time so that their players have time to get used to the altitude and for their bodies to start ramping up red blood cell production a bit. Now, less than 1% of the volume of blood is going to be the last two formed elements. We have leukocytes, L-E-U-K-O-C-Y-T-E. Prefix leuko means white. These are your white blood cells, or WBCs. White blood cells are the cells of immunity. They are the active cells produced by the lymphatic system, circulate through the body, search for all sorts of pathogens that get into you, bacteria, viruses, cancer cells, parasites, allergens, any of the nasty stuff that gets in. We have several different kinds of white blood cells, each of which has its own jobs in carrying out the immune function. Some of them directly attack pathogens. Some of them alert other cells as to where the pathogens can be found. Some of them even play the function of what we call immune memory, by which once you've been exposed to a pathogen and the body learns how to fight it, it retains that ability for the future. We're hoping we can all do that with the coronavirus once we have exposure. And then finally, the last formed element in our blood, which is unique to mammals. Most everything I've been talking about here, all vertebrates have, but uniquely mammals have a feature known as platelets, P-L-A-T-E-L-E-T-S. Platelets are little tiny fragments of broken down cells. They circulate in the bloodstream. They secrete some growth factor chemicals, but most importantly, platelets are involved in the formation of blood clots, that if a blood vessel should be broken, platelets will accumulate at the sites and sort of form what we call a platelet plug to block off the flow of blood while the body is forming elasting clots to start working on the injury. All of the formed elements are continuously being worn out and replaced. New ones are made in your bone marrow, M-A-R-R-O-W. Your bone marrow is a soft tissue inside of your bones that through a process called hemopoiesis, H-E-M-O-P-O-I-E-S-I-S, continually makes new red blood cells and new white blood cells to replenish those that have been lost. All right, folks, so I think I have told you the essential things you need to know about animal circulatory systems. You've got a basic idea what's going on. You've now heard about both circulation and about digestion. We now know how the body gets nutrients. We know how the body moves stuff around. The last part of this story of getting your body the stuff that it requires, we'll be looking at animal respiratory systems. So that will be the subject of my next lesson or two. But in the meantime, I hope you have enjoyed our talk today. I will talk to you all soon. Hope you're having a good evening.